It's very, very nice to be here. So this morning what I really want to talk to you about is to get you to think about maybe biology in a different way. I think about biology in a different way. Sometimes I barely think of myself as a biologist and a little bit more of an engineer. And I want to get you to start thinking about that from a different perspective and start thinking about what you can do with plants from a different perspective. And so to begin, we are actually going to go back in time a little bit, and probably for the first 10 minutes, I'm not going to talk about biology or plants hardly at all. So we're going to start back in 1975, when this really interesting conference took place at a conference venue called Asilomar on the west coast of the States. And at this conference, a group of scientists got together to talk about how this new technology had just been discovered, and that technology was the discovery of restriction endonucleases, so proteins that will cleave DNA at a particular sequence signature. And to them at that point in time in biology, it was so revolutionary that they could think of all these new experiments that they were going to do. They were going to recode DNA, and they started asking these questions about, now that we can rewrite the genetic code, what are they going to say? And it was so amazing that they excluded most of the press from the conference, but somehow Rolling Stone magazine was there, and for the very first time, perhaps the last time, biology was really cool, and genetic scientists were on the front of Rolling Stone. We've invited them to other meetings since then, and they don't turn up anymore. Unfortunately, we're not that interesting anymore. So this point in time passed. A couple of years later, Arbour, Nathans and Smith won the Nobel Prize for that discovery. And it's very interesting at that point to note that there was an editorial on the awarding of that Nobel Prize. And it said that biology at least was entering this new era of synthetic biology. And I kind of think about that now, because that's not even the first use of the phrase synthetic biology. I think in the 1970s particularly, or just having gone through this mid-20th century, the word synthetic, I think, synthetic fibers, synthetic fabrics had just come into being. And in this context, I think they were thinking about synthetic biology very much as being artificial. They were thinking about remaking DNA molecules in a different way. So now I would perhaps describe myself as a synthetic biologist, but in fact, synthetic to me does not mean artificial. We're not making artificial things. That's not the point. We mean synthetic from its original term before synthetic came to mean artificial, and synthesis means really expert in putting together. It comes from this Greek, synthetikos, expert in putting together. And I think synthetic biology as a field is trying to achieve not making artificial things. This would be ridiculous. We're synthesizing things, they are natural products, but more how to make the engineering of biology easier and more predictable. And it has its roots not in biology. In fact, I barely think it's a discipline of biology at all. It's really a discipline more that came out of computer science, manufacturing, and engineering. So I'll just give you a little anecdote to show you really how one of the roots and some of the leaders of the current field of synthetic biology came about. So is this familiar to anyone? Does anyone know what this is? So more than likely, all of you have got a more advanced version of this in your bag or in your pocket. This is the first integrated circuit. It was built in 1958, also resulting eventually in another Nobel Prize. And integrated circuits became chips, and they eventually became microchips. And obviously this is revolutionary. This underpins all of the electronic things that we have today. So the interesting thing about microchips is that from this time from they being invented, they started being integrated into computing. And there was a very time, probably some of you were thought about being born, and then subsequently being born and growing up, where the speed of computers was more or less doubling every couple of years. And this is because the the number of components that were integrated in each one of those chips was doubling all the time. And then quite recently, that started to plateau out. And particularly some people in computer science started to think, well, how are we going to get faster computers if we can't keep putting more components onto microchips anymore? And that one of the people that started thinking about this went in the direction of thinking, well, we could use biological organisms. They managed to put a lot more microcontrollers into a much smaller space. And then other people in manufacturing were also thinking similarly. And the thought process went something like this. What we're going to have to do in order to reach the next level of technology is to put atoms precisely where they want them. And whoever can manufacture items where every molecule is the same, where every atom in every molecule is precisely located, is going to control the new realm. And as a biologist, you think cells do that every single day. Every single day, a cell reads its DNA and it manufactures proteins and molecules, and in every one of those molecules, well, most of the molecules, I'm sure they do make mistakes, but a large number of those molecules, all of those atoms are in the right place every single time. So that makes biology a wonderful manufacturing system, except that if you really think about manufacturing, you probably think about something like this, and not something like, if any of you have done any practical sessions in biology, not something by which you're doing in the lab at the moment. So then the next step in this thought process then was, if we need to manufacture with biology, but we still want to reach the scale of manufacturing required for it to be sustainable, for it to be high throughput, for us to make consumer goods and for them to be profit making, how then can we make the engineering of biology easier and more predictable? So one of the analogies that's often made is that we can build extremely complicated cities from relatively simple component parts, and that's possible because we understand the features of all the materials that we use. They're standardized, we understand how they fit together, we understand how they behave when they're in contact with each other, and then we can make predictions about how those are going to be engineered to build something like this. And this is a nice analogy, but in no way do I want you to think that a cell is as simple as a city. It really isn't. It's a vastly more complicated thing than any of our cities are now. But nevertheless, the process of building it is vastly easier if we think about standardizing and understanding some of those individual components and how they work in combinations with each other. So the field of synthetic biology really is about applying some of those principles of engineering to biology. And there are more than just these three principles, but these are the three that I think people are working on at this point of time because our knowledge is advanced enough to work on them at this point in time. And this is to standardize things, to abstract ourselves away. So what this means is, if you think of this as an analogy to writing computer code, is that some of you may have learnt some computer codes, and more likely you've learnt a relatively high level language where at least some of that language has a grammar that's more, or a syntax that's more analogous to a spoken language, let's say. I guess that there's not very many of you that may have learnt to program in zeros and ones. But many molecular biologists still try to program cells in zeros and ones, and the equivalent of which might be DNA. So if you want to program something that's a lot more complicated, then we abstract ourselves away from working at this base level language and start working in a much higher order language. And I think at the moment one of the things that synthetic biology is doing is establishing at least a low level programming code that's a step away from that machine code of DNA. And I expect that in the next decade or so we will get better at doing that, so that we actually work in much higher level languages to be able to be more efficient at programming. And that means, in the same way that computer programmers now don't understand or read binary, but they can program very efficiently, that bioengineers will be able to program very efficiently from writing a much higher level code. So when we put these things together, then this also allows us to automate the process of engineering. And then we can push ourselves through a classical engineering cycle where we design things, build them, test them, and then we often model or simulate, and then we push through different iterations of this. So if I think what the ideal end goal of synthetic biology is, it's not really to have some ideal product, it's to have a perfect model of a cell that you can then make predictions about. You know what's going to happen, you know what you should be engineering to be able to predict the outcome, and then that model will inform the modifications that you may need to engineer into that cell to have a perfect outcome. So that's more or less what we're doing. And in our lab, we've done quite a lot of work on this, and we've established standards, particularly for working at a very, as I say, a low-level programming language. And this allows us to have standardized different parts for about which we can collect different data about how they perform in different cell types. And we start to build quite a basic understanding at this stage, but moving towards the equivalent more of where you may be if you worked in electronic engineering and you wanted to build, let's say, a toaster, and you would go buy different components and plug those into a board, and you would always expect all these components to work together. We've started to progress to this, and this allows us also then to scale down our standard molecular biology experiments so they become nanoscale and automated and more high throughput. So how does this help us in plant science? So since about the 1980s, not very long, particularly after that Nobel Prize was awarded for the discovery of restriction enzymes, people started to be able to engineer plant genomes and to be able to insert things into plant genomes. And for the most part, for a very long time, this was really just the insertion of single genes randomly into plant genomes. And this still happens today. It's still a very useful tool in biology and also in plant biotechnology. So, I mean, this phrase of genetically modified organisms might encompass that, but I don't really want to get stuck on the nomenclature of what's a modified organism and what's not. I think it's all rather arbitrary, considering the much larger changes that we've made to plant genomes over the previous 10,000 years. But what is true is that from progressing from this stage of single genes to perhaps being able to engineer synthetic gene networks, which I'm going to talk a lot more about in a moment, we need more technology. So as well as the technologies for just being able to insert things into plant genomes, we also need to be able to modify and rewrite the basic code that exists. And as we increase in complexity, then it becomes more important to be more predictive, to have a better understanding of the system so that all of these things work better. So we hope by sometime in this century, we'll be getting away more from the single gene randomly inserted and more to be able to have a more consciously and more rationally engineered plant at the end to solve some of our problems. So one of the things that we are interested, or one of the things that I'm very interested in solving at least, is since we've had transgenic technologies, it's been relatively easy to insert single genes, and it's been relatively easy to knock down or to repress the expression of single genes. And this is very useful when you are looking at plant traits that are controlled by a small number of genes. But a large number of plant traits are not controlled by a single gene. Instead, they're controlled by a network of genes that all work in coordination with each other. So, for example, in Ottoline's talk yesterday, she talked about plants' responses to environmental nitrogen. And she mentioned that in response to environmental nitrogen, the plant will remodel, for example, its root system architecture, the amount of vegetative biomass, even the time that it flowers. And yes, there are these master controllers that do this, but at a basic sense, for a root to grow, there are a lot of individual processes that have to take place. So these individual signals then get deconvoluted through these networks. So this is a transcriptional network for a plant. This is just an example of one by which you may have something which senses an environmental signal, and then it will inform it will start upregulating or downregulating other pathways by acting directly on those genes to up or downregulate them, and then this will in turn make cascades of genes react to, for example, remodel that root, remodel the way it transports nitrogen, the way it is making amino acids, the types of organs that it's going to make in response to those things. So each one of these is for a transcription factor in this network. So this is a type of gene that will bind to the regulatory region of a different gene and modify its expression level. So each one of these lines interactions can be thought of as the interaction between a protein and a DNA sequence. So what we would like to do is understand how genetic and epigenetic variation in these non-coding regulatory regions of multiple genes can be re-engineered. So the other thing about crop plants today is that domestication, this process of domestication, which Ottoline mentioned yesterday, with this difference between, for example, teosinte on the one hand and sweet corn today, in that process of domestication, we fixed very desirable traits, almost always those reflecting yield, but we reduced the biodiversity in our breeding population. So in a wild species, you will have a lot of genetic variation. As plants were domesticated earlier, we lost some of those things because we were just selectively inbreeding for the traits that we wanted. And by the time that we get to our modern varieties, we have relatively little genetic variation in those pools of plants that we can breed within. And all crop improvement practices today are really trying to capture within these elite high-yielding lines the genetic variants that control desirable traits. So we've already said that we would like to have more variation in these non-coding regulatory regions, but we have a paucity of those in our breeding populations. We've also said that these networks are really complicated. It's not just a matter of knocking out one of these genes, because the consequences of these are going to be quite wide-reaching. You're going to remodel your plant in ways that are sometimes unforeseen, and they're going to have effects outside. So if you just want to look at root development and you start messing around with something here, you're probably going to mess around with all these different parts of the plant and end up with a plant that wasn't quite what you were looking for to start with. But these networks in themselves has a robustness attached to them, and they also have key regulators attached to them. And while we can reconstruct these networks from looking at how the transcripts at the plant respond on a global level, it's quite difficult to do experiments to understand these networks unless we can individually perturb these connections and rewire them. So what I've been thinking about for quite a long time is how we can use biotechnologies to rewire gene expression networks to engineer these complex traits that are otherwise very complicated, somewhat retractable because of their complexity and the disadvantage of not having that much variation across our regulatory regions to play with. So to do this, I think we need two or three bits of tools. We need tools that will allow us to make mutations in non-coding regions that will specifically disrupt connections. So this may involve, for example, knocking out or making a mutation in this particular region by which the protein binds to this DNA, but also understanding exactly what that's going to do to that connection. We also might want to make, for example, here a synthetic connection. So here I've put in an example of a feedback. So we have maybe a synthetic transcription factor which will bind to the promoter of this master controller and inform it about the state of something much further down. And this synthetic transcription factor will be driven by a synthetic regulatory sequence that only responds here. So we're driving feedback and thereby increasing the robustness of this circuit. So hold that thought. We're going to switch gears a little bit away from that kind of farming and talk about farming with the pH and producing pharmaceutical compounds, and I'm going to bring those two back together. So plants, as well as feeding us, make a whole range of amazing compounds. So here's just an example of some of those. You will see in here things that are used as drugs, things that are used as flavors, things that are used as scents. But often these are found in hard to cultivate species. Some of these individual molecules are difficult to purify, and some of them are also difficult to chemically synthesize. Now I know some really, really good chemists, and there's almost nothing that they can't make, but that doesn't mean that they can make it cheaply enough or easily enough to be able to make it as a consumer product, or even as a drug, which is why some of the drugs that we use are so expensive, is because they are in these rare species. So in synthetic biology, I think particularly there's been a kind of a movement away. So we take a product like indigo. Anyone wearing blue jeans today, they probably dyed with a indigo. And if you had bought them maybe 100 years ago, they may have been come from an indigo that was extracted from plants. If more recently in the 20th century, late 20th century, indigo has mostly been chemically synthesized using aniline as a starting point to build this molecule around it. And then more in very, very recent times, and probably not a product that you can buy yet, people have isolated the genes from this indigo plant that makes it and have managed to be able to recode these. So here you see some E. coli colonies on a plate, and they are making indigo, and they have the color of indigo. So here we have this heterologous biosynthesis of indigo in a different biological chassis, and hopefully moving to a more both humanely and less chemically damaging synthesis pathway. So another example might be vanilla. Vanilla comes from an orchid. It's native to Mexico, but most extensively cultivated in Madagascar. For many, many years, it has been extracted. So many of you may be familiar with vanilla extracts. It's also possible to chemically synthesize it. There's a few different routes of doing that, but usually it comes from these two molecules as a starting point. And then more recently, then there has been an elucidation of the biosynthesis pathway from orchids, and people have taken the codon sequences for those genes and reprogrammed them largely into yeast. And different companies are using different starting molecules. I think the most comprehensive one is a company called Evolver, and they start with glucose that comes from comes from corn, sorry. And they feed this, and the yeast ferment this until they make more vanilla. So here we have a heterologous biosynthesis pathway. So in a project that we're working on at the moment, both of the extraction and chemical synthesis routes have somewhat failed. And what we would like to do in our project is to make insect sex pheromones. And the reason that we'd like to make insect sex pheromones is as an alternative to broad spectrum pesticides. So a lot of you will probably have seen in the news that our insect populations and consequently our bird populations are reducing. The number of bees that we have, of pollinators, are falling. And some of the culprits of these are the broad spectrum pesticides that we spray on our crops to keep them pest free. So an alternative might be to try to use a more specific control agent. And in this case, we're thinking of using insect pheromones. In fact, they are already used quite successfully. But insect pheromones, they're nicely specific just for that species. You don't have to use them to kill them. You can just use them to confuse the males so that they don't find the females and they don't lay their eggs and larvae that will consume the crop. But obviously, we can't really purify the pheromones very easily from the insects. Some of these molecules are incredibly complicated, and it's just not possible. So, for example, this particular pheromone, which is made by the citrus mealybug, we think there's really only one enzyme that turns this from a relatively simple chemical into this rather complicated chemical. But doing this by chemistry would take 12 or 13 different steps, all of which are reasonably expensive. But what we are able to do is to identify the genes from the insects, pop them into some plants, and we can then have a plant that makes these exact same pheromones. So here we have this is a mass spec trace of a plant. This is a control plant, and here we have our so-called sexy plant that is making these insect sex pheromones, and you see the two peaks, and these moths do respond to that plant. So this is in the newspapers very recently, and this has been dubbed as sexy plants to be on track to replace harmful pesticides to protect crops. So as I said, most of the sustainable bioproduction that has been done to date has been done in microorganisms. So we see these large fermentation platforms. So this factory is fermenting yeast, and in this factory they're actually using, they're feeding sucrose from these sugarcane fields that surrounds it to make this molecule, which is known as trans-beta-farnesene, which is used in emollients and medicines and many other things. So this is fantastic, but I can't help think, as a plant scientist, that it's amazing, and this production factory is also in Brazil, that we used to have a rainforest, and now we've got a sugarcane field, and it's fantastic that we can make this molecule. I do question it being sustainable manufacturing. And I think really the sugarcane's probably making an abundance of really interesting molecules, and it seems kind of a shame to just rip out the sugar to turn it into a single one. And I think actually plants do more interesting things on their own. So we would like to use plants themselves as a manufacturing platform. As Ottoline was saying yesterday, plants do this incredible chemical reaction of turning thin air into molecules that you can touch and feel, and that can cure diseases. But while we have a rather sophisticated toolbox for engineering microbes, we have a rather poor toolbox for engineering plants. So when we try to express some molecules in plants, the plant responds in all kinds of ways that we don't really want. So we may be trying to make a simple molecule like this, but the plant recognizes this because it has this really rich metabolism, and it immediately turns into this. Some of the proteins that we make elicit a response, it recognizes it, and it starts to produce peroxidases. Peroxidases themselves trigger this defense pathway, and you start to see cell death where you're trying to make it. Some of the plants that we use, which often, for reasons that I'm not going to talk about today, are often related to tobacco, make a whole bunch of things that we don't want them to. And to deliver DNA into plants itself, we're often also using a microorganism, agrobacterium, and this is recognized as a pathogen, and therefore the plant responds to it slightly as a pathogen. It's not going to cause a full disease because we've modified that so that it doesn't do that, but the plant still recognizes it a pathogen, and it mounts a transcriptional response to that pathogen that kind of modifies its entire metabolism. That again makes it quite difficult for us to ensure the yield and purity of the things that we want to make. So we would like to do a few things. We would like to rewire networks. We would like to perturb these specific connections. We would like to engineer our plants to modulate plant responses to foreign molecules. We would like to drive carbon flux into our pathway to increase our yield and purity. We would definitely need to, if we're going to do this heterologous expression, to regulate the relative expression of these suites of genes that we need to make these complicated molecules. And then we also need to be able to make these new connections. So what brings these things together is a bunch of technologies that we need. We need to induce specific informed mutations in coding sequences and in regulatory sequences. We need synthetic transcription factors to control the level of endogenous genes, either here or for here. And we need synthetic regulatory sequences that respond to external signals or to specific endogenous transcription factors. So they're the kind of things that we are working on at the moment. So just to orientate ourselves, so far today we've talked about, I think, synthetic biology, which I describe really often as a biology from the perspective of being an engineer. We've talked about really how applied science is much more than just agriculture, or it can be. And we've talked about how a better understanding of gene regulatory sequences can be used to engineer plants for both farming and perhaps farming. So in the last little section of the talk then, I just want to talk about some of these tools, these genome engineering tools, and how we can use them to induce specific mutation, build synthetic transcription factors, and also how we're decoding pathogens to start building synthetic regulatory sequences to respond to things that we would like them to respond to. So no doubt some of you have heard of CRISPR. CRISPR is a tool, if nothing else, and I'm not going to tell you in detail how it works. I'm not going to tell you where it came from or what it does. All I'm going to tell you about is what we use it for. And in its most basic sense, just harking back to what we spoke about at the beginning of this talk, it makes breaks in DNA. So as the restriction endonucleases that were discovered in the 1970s will make a break in DNA at a specific motif, a sequence motif that it recognizes, what genome engineering tools we are able to do with them is we're able to reprogram them so that we decide where they cut. So instead of having a protein that will always cut to ATTGC, we can say, well, I want you to cut here, I'm going to recode this, and I'm going to send you to make this double-stranded break. So what happens when you make a double-stranded break? Well, if you're just making it in your nice little test tube where all you've got is your DNA, and you cut it, nothing will happen. If you're also still in a test tube, but you're adding another enzyme, DNA ligase, then that will stick those two molecules back together. But in cells, in plant cells, in any kind of cells, they have their own mechanisms of mending these double-stranded breaks. And they can be repaired in a few different ways. And I'm not going to talk about the mechanisms where they do them. I'm just going to talk about more or less the outcomes in terms of engineering. So most of the time, if you make a break, it's just going to be repaired. And a lot of that time, that repair is going to be absolutely perfect, because otherwise your cell would break all the time. DNA gets broken for lots of different reasons. It encounters mutagens. Even sunlight is a mutagen. It makes double-stranded breaks. Every so often, it's going to be repaired imperfectly. And this may be a small insertion, deletion, or inversion. And this, for us, if we've invoked this ends up as being a targeted mutation. We can also try to provide a repair template that will be inserted into there, so we end up with a targeted insertion. Sometimes we can try to leverage a different repair pathway of homologous recombination, where it uses a very similar or identical DNA molecule as a repair template with which to repair it to. And this, you end up with a much more precise mutagenesis. So here we've encoded the differences that we want, and we end up with something more like a recoding. And this I would call true editing. Alternatively, you can flank your sequence that you want to insert and try to drive homologous recombinations and end up with a targeted insertion and a more precise entry. So we've been able to leverage this for several different things. So just showing you how the technology works, one of the experiments that we did really early on, so this is in a plant called Brassica oleracea. It's an oilseed plant. We targeted a gene, GA4. It's in the gibberellin biosynthesis pathway, so it's knocking out the production of a plant hormone. In Arabidopsis, this is already known. If you knock this out, it gives a dwarf stature, and it changes the morphology of the seed pod such that there's reduced dehiscence, so the seeds release a little bit more easily. So we programmed, in this case, a CRISPR construct that would induce a mutation into this gene. So on the top here we have the wild type, and then here we're seeing 2 generations of plants, after which we have an induction. We see the same expected dwarf stature as we would expect to see from knowing what this gene does in Arabidopsis. We're also able to segregate out this initial transgene because it's not genetically linked to the mutation that we have caused with our programmed enzyme. So we end up with a plant. So these plants here now only have this little lesion in their gene, and otherwise they are substantially genetically similar, not completely, because there are always changes between individuals, to the plant that we started with. We're also able to drive precision editing using a DNA repair template. So this is in wheat, and we are targeting a gene that exists on chromosome 6. So wheat is a polyploid. That means it has multiple copies of its genome. It actually has 3 copies of its genome. So there are 3 copies of chromosome 6 in a diploid cell. And here we are able, in this case, we're using zinc finger nucleases. We've targeted in the line above. It doesn't matter that you can't see the details here at all. Here, I just want to show you that we've made a single break, and we've inserted this piece of DNA here. And these red blocks here are just showing differences from the wild type sequence. So we've recoded the last part of this gene. So in this case, this is just inserted into it. So if we look then at this location on a gel, we see a few different copies. We see the larger one, the slightly larger one, which has this inserted, and we still see some of the wild type one, and this is because we didn't manage to get an insertion in all 3 copies. Alternatively, we could make 2 breaks here, and we have inserted this and done a complete allele replacement of this particular gene. More recently, we've been able to avoid putting DNA in altogether, and instead of making a construct that then expresses the programmable nuclease, we can make that programmable nuclease first and deliver the protein, and in this case the RNA that guides it to the place that we want to cut. We can hook this up to a YFP, or fluorescent protein, so that we can see what it's doing. We can deliver those to plant cells. We can test that this complex is working as it should just on a double-stranded template, and here it works very much like a restriction endonuclease. It just cuts things up. And then if we deliver it into the cell, here we've delivered 2 to make a small deletion, so we see this fragment start to drop out where we've managed to mutate this. So now we have the possibility to be able to model genomes without inserting something else directly into the genome. So I said that the difference between a genome engineering tool and a classical restriction enzyme was the ability to program it. And this is because it has this programmable DNA binding domain. And we also talked about transcription factors as really being proteins that bind to DNA. So you can modify these tools for engineering and take away their ability to cut DNA, and instead you can add a different protein domain so that they can make a different change. So in this case, we can hook up an activation domain, for example, to activate or repress transcription, and then we end up with a programmable transcription factor. So we have our DNA binding domain, which is exactly the same as one of those programmable nucleases, but it doesn't make a break, but it will still recognize where we send it to, and it will, when we express it in the cell, it will drag along this activating domain to it, and in this case then we upregulate its target gene. So in this case we are in fact targeting a gene that induces cell death. So where we've inserted this into our plant cells, we start to see, it's quite a little bit hard to see on the screen, but you start to see that those areas of the plants are doing. These are actually experiments done by undergrad as part of an iGEM project a few years ago. We can also then start to make more complicated arrangements of those. So here we have our same activators, and we've made a logic gate. So logic gates are often used in engineering. And here we have a NOR gate. So if there is an input from either A or B, then our output is 0. So input from 2, sorry, I've called them A and B and 1 and 2, this is confusing. So if we have an input here from A or B, or from both of them, we have an output of a 0, and if nothing's happening with either of those, we have an output of 1. How this might look in a genetic circuit is, here we have our same activating domain. So here we have 2 different binding sites in this synthetic promoter region, and we use this to express this particular protein, and this protein itself is a repressor of transcription, and this binds to a different sequence. So if neither of these are present, then this is not activated, and you will see GFP fluorescence. If either one of these is activated, it will bind to this. This will start making a repressor, and it will repress DNA, so repress the expression of GFP, and then you will no longer see this. So here you start to see ways by which you can build combinations, build sequences of DNA to start to build more complicated switches. So if in this alternative or this alternative, I want this particular outcome. So you can transfer some of this logic into a single sequence of DNA. And this is where it's very helpful to stop working at this primary 0, 1 ATGC code and start working at this slightly higher level language of operators, because otherwise these concepts become very difficult. So often plant biologists for many, many years utilize regulatory sequences from pathogens to constitutively overexpress genes, or at least they tell themselves they're constitutively overexpressing, and mostly they are, but not always. So usually a very simple experiment, someone wants to overexpress something in a plant and see what it does. They take regulatory sequences, often from a pathogen, and they flank them either side of their particular gene of interest. And they pop it in the genome, and it upregulates. And for many, many years, people have done this, and yet we know relatively little about these regulatory sequences. And I find it absolutely fascinating that these pathogens, and lots of different types of pathogens, have evolved to so specifically utilize these regulatory mechanisms that are within the plant, and how they do this. So to do the last of the trio of things that we need to do, we've been trying to decode how these work so that we can start rebuilding different regulatory sequences to respond to the things that we want them to respond to. So new sequencing technologies and bioinformatics tools have started to allow us to understand some of the old data. So really old experiments that people used to do is they used to take regulatory sequences and they used to start deleting little bits as best they could at the tools that they had, and then they could say, hey, at this point we get a 90% drop in, we still have 90% drop in activity, but if you delete this section, it goes all the way from having 70% to 20%, so this bit must be really important. But at that time, we didn't really have the tools that would allow us to look at the motifs, the proteins that were binding to them, what was happening with that DNA to understand them. So now we do have new sequencing technologies that tells us about those things, and we can start seeing some of those motifs and identifying the specific proteins that bind to them. So this allows us to map these candidates. So this is a transcription factor binding site or motif, and we just map this onto a deletion analysis of a promoter. And these deletion analyses were done maybe 30 years ago, but now we can start to understand what it is that they were doing and what's happening in this DNA sequence. So then we've also developed a rapid method of being able to quantitatively characterize what's happening in these promoters. So we can test either variations of these native promoters, or we can look at, we can start with a random sequence. So we have a library of these that we know give no transcriptional output unless they are attracting a transcription factor. So to this then, we can start either mutating our native sequence, or we can add binding sites into this, and then we can hook this up to a reporter, and then we can always express that as a ratio of something that we know expresses the same in every cell. So going back to our pathogen promoters, what we've been doing is we've been taking some of these binding sites, we've started to make rational mutations to try to understand the points where those DNA and proteins interact with each other, and then by making these tiny variations, we can start to tune the effect of those. So some of these have very little effect, and some of them just by making a very small change. So here, if you look at this, these few changes here, we start to have quite a big significant change just by altering one or two base pairs, which is only possible to do because we know what to alter and we know what's happening. We can also then harvest all these things that we start knowing about these transcription factors, and we can start building synthetic promoters. So here we just have a library of different things, and we have our same blank DNA sequence, and into that we've just dropped a range of different candidate transcription factor binding sites, and we see how they express in different, at this point this is just all in one cell type, so we can just compare them with each other. We can then start putting them together in different combinations, understanding the types of proteins that they bring together, how they interact with each other, what that does to change the DNA molecule, and what that does to change transcription. In all of these cases, we know what that transcription factor is, we know where it expresses in the plant, we know what it's responding to, and now we're beginning to have an understanding of how the affinity of that protein to that DNA sequence then affects the amount of transcription. We can then start shuffling all these domains because it's not just a single thing that works, it's often how they work in combinations with each other. And we now know that the relative positions of these, sorry, I'm again switching nomenclatures, these transcription factor binding sites or cis-regulatory elements, how they affect function, and we can start making suites of synthetic promoters then that we can tune. So, at this point of time, and in no way am I saying that we have solved this problem, but we are getting to the point where right now we are preparing to get ready to start rewiring transcriptional networks. So we have tools that I've showed you that allow us to make mutations in non-coding regions to disrupt or strengthen different connections, perhaps by modifying just those specific individual bases that will increase or decrease the affinity of a protein to a DNA binding site. We can make synthetic transcription factors, and we can target where they will bind, and we can start making synthetic regulatory sequences that respond to particular transcription factors. And we can hook these together and do these not just for single genes at a time, for many of them, to start trying to understand some of these more complicated things. So I've just got one last thing left to say, and that is to go back to that meeting that happened at Asilomar. I was lucky enough to be there myself at a not too dissimilar meeting a couple of years ago. And while I was there, I read that whole Rolling Stone article, which is actually quite extensive. It's about 6 pages of Rolling Stone explaining how biologists behave at the beach, largely. But there's this lovely excerpt from it. I'll read it to you. It says, at one point, for example, a tan young Southern Californian clambers on stage holding a 3-foot-tall weed freshly harvested from a Monterey roadside as a colleague passes out similar plants to each row of molecular biologists. It's a legume, says the plant specialist, and if one shakes the dirt off, it's possible to see the tiny root nodules. Tiny nodules on the root structure that fix nitrogen, adapt it to forms useful for living organisms directly from the soil. Not a bad trick, since most world food crops are not legumes and thus require doses of artificial nitrogen fertilizer, created in turn from a commodity somewhat scarcer than dirt, petroleum. Clearly, if one were able to isolate the gene that teaches nitrogen fixation and ally that, say, with a wheat plant, then one might have one hell of a food crop. So unfortunately, I wasn't able to find out who this tan young Californian was, but obviously he's going to be much older now anyway. But I did go looking around. I did go looking around the shores of Asilomar, and I'm pretty sure that this was probably the weed. It's a legume that grows quite extensively around there. It's this beautiful coastal bush lupine, and I feel like this is probably what he plucked out the ground to show to all his colleagues, most of whom were medics and were not that interested in plants, to show how amazing it could be. And obviously it is not as simple as finding a single gene to teach nitrogen fixation. It's a network of genes, it's a relationship between two organisms, but I love that long ago, as soon as people had this idea for a new technology, they were thinking about these really ambitious things that they could try to solve. So I just want to finish with that now we are a little bit better at writing the genetic code for you to think about what you might like to say. Thank you.